Swayam Prabha. Digital India. Educated India.
So, from this lecture onwards we will look at more advanced topics in NMR, and we will start at first with how we can use the understanding of the different spectroscopy techniques in NMR and apply it to the structure determination of molecule. Specifically we will look at a peptide, because peptide molecules illustrates the complexity involved in determining structures of biomolecules or molecules in general by NMR. So, let us look at that first. So, we will see now, as you can see, this is a general flow chart which typically people use when we solve structures by NMR. First thing is that you need have a sample. So, you need to purify your sample, and as we saw, I mean, the purity is a very important parameter when it comes to looking at structure determination of molecules. Typically we say that it should be more than 95% pure. The reason for this is that the impurities present in your sample will have extra peaks or correlations in your 1D and 2D NMR, and that can lead to ambiguity or doubts in solving structure. So, therefore, if it is more than 95% pure, it is very unlikely that the small impurities will give any strong correlations in the spectrum. The second is, once you have the sample, we typically take about a millimolar sample. This is for case of biomolecules or natural products, but if you have an organic synthetic synthesis molecule, you can look at higher concentration, you may go even up to 10 millimolar. But in a standard NMR spectrometers nowadays, 1 millimolar is sufficient for a good spectrum to be collected. So, you put the sample in the magnet, and then we record a series of NMR data. So, this is typically 1D NMR, 2D NMR, and in case of biomolecules like proteins, 3D NMR, but we will restrict in this course only to 1D and 2D. Now, once you record the data, we have to analyze the data, we have to process the data, and we have seen that how do we process data and how we analyze, and based after the analysis, what is the objective of the analysis? The objective of the analysis is mainly to achieve the third step, which is resonance assignments. Means every peak in your spectrum, be it 1D or 2D, the goal is to assign this to a particular atom in the molecule. Okay. So, the every peak should be assigned. Now, once the assignments are done, then we go on to structure determination. So, remember, structure determination here, what I mean is not just simply the structural formula of the compound, like we saw in the case of 1D and 2D NMR. We looked at few examples where we had only the chemical formula, molecular formula, and we tried to figure out the different functional group connectivity and look at the structural formula. But that is not what is meant here. What is meant here is the complete 3-dimensional structural information, and that comes from the very important experiment called NOSI or ROSI, because remember we saw that each peak in a ROSI or a NOSI spectrum gives the information of the distance between atoms. And what is 3-dimensional structure? It is nothing but a distance information between different atoms in 3-dimensional space is what we are looking for. So, that is what is meant by structure determination. So, these are basically the steps involved in going from sample to structure by NMR. So, where do the different experiments that we learnt comes into picture? So, it comes into picture in the following manner. So, let us say that we have the molecular formula of the compound. How do we get that? That is by different approaches like chemical or elemental analysis. It gives you the amount of percentage of nitrogen, oxygen, carbon, hydrogen, etc. And mass spectrometry gives me the mass of the sample, and combining these two information we can generate a possible molecular formula. and that is what is done in chemistry, and we will not go into detail here. Now, given a molecular formula, this is the examples which we have seen, we can first initially characterize the molecule by simply 1D and carbon and proton NMR, and at this step all you are doing is you are simply recording the spectrum and then identifying what are the peaks, how many peaks, are they commensurate, means are they equivalent to what you expect from based on this formula, and so on so forth. So, this step is not going to give you structural detailed information and is only meant for just initial characterization. Once you have done that, we need to now accomplish or achieve chemical shift resonance assignments. For that, we need to go to higher dimensional, that is 2D NMR. So, for small molecules, organic compounds, typically this set of experiments are sufficient to establish the resonance assignment. So, this is what we all of these experiments we have seen in this course. If you go to peptides, which is a link of amino acids, again this sort of experiments what we discussed also will be sufficient. In fact, just 2 dimensional experiments, NOSI and TOXI or TOXI and ROSI is sufficient to establish or accomplish this step of chemical shift resonance assignment. Then once you have done that, the next step is to do now the full fledged structure calculation, that is determination of 3D structure using NOSI. So, the information of NOSI is used and the cross peak intensities are converted into distance information, and those distance values are given to a program, computer program, which carries out what is known as molecular dynamics calculation, and the output of that is the final structure. So, this is typically for peptides. When you go to proteins, which are bigger molecules, there 2D NMR is not going to be sufficient. We have to go to even larger high dimensional spectroscopy, that is 3D NMR, and not only that, we need to do special sample preparation by doing what is known as carbon 13 N15 labeling. But we are not going to go into details of that particular aspect. So, the whole course is only focused on small molecules and peptides. So, now we will focus on how we can assign and get the structures of peptides with 2D NMR. So, this is how the process goes on. So, first thing is you need to have the exact peptide sequence with you, that is you should know the sequence, that is how the amino acid sequence is there in your sample. So, let us say you have this, remember there are 20 naturally occurring amino acids in biology, in nature, and we are also, they can be unnatural amino acids, a very synthetic organic compound can be inserted, but all of these are connected to each other by a peptide bond. So, what is a peptide bond? This is well known to all chemists. Basically, a peptide bond is looking at this kind of a connection. So, this is one amino acid, I will note it as AA, a second amino acid, and these two are linked together by the peptide bond. So, this is what we are going to focus on in this next few slides, or this part of the course, where we look at the structures of peptides. So, this is the peptide bond, but you can have unnatural amino acids, but we are again will take an example of a natural amino acid containing a peptide containing natural amino acids, and there are 20 of them. So, this could be the sequence example, but remember you can also have the same amino acid repeated multiple times. So, look at this example, I have a peptide which has alanine, arginine, asparagine, alanine, so on so forth. So, alanine has come twice. So, there is no limit or no restriction as to know how many times the same amino acid can repeat. So, this is like a polymer. So, we are looking at a biopolymer. Okay. So, this is where the complexity starts. If you have multiple amino acids of the same type, then the spectrum is complicated to interpret. So, let us see how that can be accomplished. So, what are the two goals in our whole project now? The goal is to assign the chemical shift of each peak of the spectrum which we get in the spectrum to the respective amino acid in the peptide. Okay. So, for example, your alanine which has a CH alpha and beta, which we saw in the example in the last class, then there it has alpha proton, alpha beta proton, alpha carbon, beta carbon. So, I have 4 carbon and proton, and of course, you have the carbonyl and amine, which we will not consider right now. We are looking at only the alpha and beta carbon proton, and that alpha carbon, beta carbon protons, where do they come in the spectrum, what peaks chemical shift are those things have to be assigned at the first step. Then once you have assigned the chemical shift of every peak in the spectra to the respective amino acids, then you can go ahead and start looking at the structure. So, structure means there are 2 types of structures in peptides and proteins. We say one is secondary structure, that is alpha helix beta sheet, and the final structure is what we say is 3-dimensional structure of the peptide. So, this is the basic requirement or the objectives in looking at the structure of a peptide. So, when it comes to resonance assignments, there are 2 steps involved, or it can be accomplished in 2 steps. Number 1 is that given the spectrum, find out how the resonances of each amino acid. Okay. So, let us understand what this means. It means, again let us take an example of this peptide shown on the top, alanine, arginine, asparagine, alanine. So, each of these amino acid is now a spin system, because remember, because of this peptide bond here, the connection between this amino acid to this amino acid by J coupling is broken because of this intermediate long distance. So, the proton here is very far away from proton here. So, there is no coupling between them. So, basically we can say that each amino acid is like an isolated spin system. So, if I have 4 amino acids in the peptide, I can say it is a tetrapeptide, means there will be 4 spin systems in the spectrum. Okay. So, in a toxi spectrum example, you expect to get 4 different spin systems, but alanine is twice here, 2 times. So, there will be 2 spin systems for each alanine. So, the first step is that given this spectrum, we have to identify the spin systems. Okay. So, we have to say that if you recollect in the last class, we saw the HSQC toxi experiment, where we saw that the valine spin system has a set of spins connected to each other in a toxi. Similarly, leucine had another set of peaks connected to each other. So, we could differentiate or separate or distinguish valine from leucine spin system by toxi. Same thing has to be done in this case. So, once you have identified the spin systems, then you have to find out which spin system comes in which sequence or order. So, look at this again. In this sequence, my first amino acid is alanine, and second is arginine, third is asparagine, and fourth is alanine. So, that means the 2 alanine spin system which I would have got from my toxi, I should now then decide which spin system comes this side, that is the 4th one here example, and which spin system alanine comes in the beginning. And that is not easy by just looking at the spin system, because the spin system as such does not tell you anything about its location in the sequence. It only tells you there is an alanine present, or there are 2 alanines in your sample or in your peptide. It does not tell you which alanine spin system comes the last, or in this case the 4th, or which spin system comes here. So, that kind of arrangement of arranging the spin systems in the right sequence is called as sequence specific resonance assignment. So, this is a very important step. Only when these 2 steps are completed, then only we can say that a molecule or a peptide has this goal is achieved, that is the goal of chemical shift assignment is achieved. So, then once you do that, then comes the next step of secondary and tertiary structures. So, let us see how we can achieve these 2 steps shown here, that is of assigning spin systems and then followed by sequence specific resonance assignment. So, this is example again of a peptide or a protein sequence which runs like this. So, typically we denote this side as N terminus. So, let me write that down. So, So, we say this is called N terminal residue and this is this side is C terminal. So, this is the notation which is used in NMR and therefore, we will use that notation to talk about the assignments. So, now if you see generally if you go to each amino acid and this is a dipeptide will be like this. So, let us look at this carefully. What we are seeing here is the individual atoms and you can say this is N, this is one amino acid, this part when shown separated by a dotted line. So, this portion is one amino acid and this portion is another amino acid. So, if you go by this notation we say I means this amino acid, the first amino acid, first in the sense any amino acid we will use the word I, arbitrary I, and then the one previous to it, that is before it in the sequence will be now called I minus 1. Okay. So, when I say when in the next few classes we will discussing I minus 1 like that, you should immediately it should come to your mind that I means the amino acid, any amino acid which is self. So, we use the word self. So, let me write that down. We say this is self and we say this is sequential. Okay. So, we use the word self and sequential to denote these two different amino acids. Okay. So, this is general, this is a peptide bond which is basically linking, connecting the two amino acids. So, in the protein and peptide NMR jargon terminology, we have to define some more terms. First is the backbone atoms. So, this zigzag chain running through the sequence, that is it will go this side and which has H alpha including the H alpha, we use the word backbone atoms, and the remaining which is this beta onwards. So, you will have gamma, delta, epsilon on every amino acid, that portion of the amino acid is known as side chain. This two differentiation is done because in peptide NMR and protein, in specifically protein, not so much in peptide, there are different set of NMR experiments which are particular to only the backbone atoms or those which give correlations for side chain atoms. So, there is therefore a segregation in terms of the nomenclature and we distinguish these two sets of atom, backbone and side chain. So, now let us move ahead. That now suppose I record a 1D spectrum of a peptide. So, what kind of spectrum I expect to get and what are the different chemical shift values. So, this is just a schematic, down the line we will see a real NMR spectrum of a peptide 1D NMR, but right now this is a slide just telling you the rough positions where the different functional groups in the peptide will come. So, let us start from this end, the 0 ppm. Here comes the methyl, if you recollect, the methyl peaks comes here and the aliphatic portion, it is the gamma, beta, delta, hydrogen carbon atoms. So, hydrogen atoms, they come in this color, this position for every peptide. So, this one thing you should keep in mind, this whole picture which is shown here is conserved, means same for every peptide. Very rarely you will get unusual values. Of course, this is for naturally occurring amino acids. It may happen for an unnatural amino acid or some special amino acid, it may deviate from this zones or this demarcations, but by and large for all peptides you expect to get correlations or peaks for different functional groups in these zones which are marked here. So, between 3 to 5 ppm you typically get peaks for all amino acids which have the H alpha of all amino acids, and then comes the water. So, typically peptide is a biological molecule and same with protein. So, we record those spectra in water or deuterated deuterium, that is D2O. In that case it is can come in the water comes at this position around 5 ppm. Then between 6 and 7 or 8 ppm comes the protons from the aromatic amino acids. These are typically phenylalanine, tyrosine, tryptophan, histidine, and these are the 4 amino acids in protein. So, this is similar to what we normally see in organic compounds, that if you have an aromatic spin system, we get peaks somewhere in 6 to 8. Amides, that is the CONH, the peptide bond, the NH of the peptide bond also comes in these particular regions, and then comes more special like imines and so on, but our major focus now is only focused on these 4 functional groups, that is amides, aromatic, CH alpha, H alpha, and the remaining aliphatic side chains. Okay. So, this is important to know what peaks comes where for resonance assignments. So, now let us look at as kind of a pattern, peak pattern for amino acids. So, again remember we are going to restrict our focus only to the 20 naturally occurring L amino acids. L amino acids are what we see in nature in all peptides and proteins, and therefore, we will focus on those 20 amino acids. So, we will look at a few examples of what is a peak pattern. So, now given that we have 20 types, now we have let us look at how an alanine cozy pattern, this is proton to proton cozy will look like, and how a peak pattern of some other amino acid threonine will look like. So, this is shown here. Alanine is very simple, and this is what we see in many textbooks, and that is alanine alpha, and we have already seen this gives a correlation to the beta proton. Okay. So, remember this and this axis both are hydrogens. So, beta proton is coupled to the alpha proton by 3 bond J coupling. Similarly, we can see for threonine you get again 3 bond correlations proton to proton. There are 3 protons here, one is alpha, beta, gamma. So, alpha goes to gives correlation to beta, beta gives correlation to gamma. Okay. So, similarly for many amino acids like this. Now, let us see how a toxi spectrum will look like for let us say some spin system. So, remember whenever we talk about toxi, the moment we talk about toxi, the word spin system comes into picture. So, as we discussed, each amino acid in a peptide is or not coupled to the neighboring amino acid by J coupling. So, therefore, you can say that they are isolated spin systems. So, example in a valine, this amino acid, I would expect correlations between only this proton to this proton and all of them, but not to any neighboring amino acids. So, this is what is shown here. You can see that this diagonal, this is a toxi peak, and you can see the correlation from toxi NH, this is from starting from this NH to the C alpha. Remember, this is 3 bond J coupling as shown by this arrow here, and then from this proton H alpha you see correlation to the neighboring the side chain aliphatic spin system. So, to beta to gamma 1, gamma 2. Now, which gamma is which proton we do not know. So, that we cannot distinguish at just by Stone's toxi. So, we will just call arbitrary one gamma as gamma 1 and another gamma as gamma 2. So, this is known as stereospecific prochiral protons. These prochiral methyls right now we cannot distinguish, but we will say there are 2 gammas. So, this is the pattern what is shown in this picture here for a toxi spectrum of valine. Now, those which are in the red color are the cozy peaks, which are only next neighboring 3 bond away, but the green peaks are one for a toxi. So, this whole spin system is now joined or connected to each other, and that is how a toxi spectrum should look like, and this is what we have been seeing in the toxi part of the course. So, typically when we go to peptides, this pattern what is shown in this green color oval circle is what we look for. This is a very interesting pattern. This is called a fingerprint region, and you can say we will show in the next slide, we will see in the next slide that all the 20 amino acids have a unique fingerprint pattern like this. So, no two amino acids are similar. Of course, if you look at aromatics, yes, a family of amino acid will look similar, but they are very well interestingly they have very unique fingerprint. So, let us see that in the next slide here. So, what is shown here is on the x axis is just the different amino acids, the 20 amino acids are listed here on the top, and on the y axis is the chemical shift proton values, but only shown from 0 to 5. So, what we are trying to see here, as in the previous slide, we are looking at this portion here shown by this green arrow, green circle, and this portion if you see, it is only the aliphatic portion of the amino acid, and that goes somewhere from 0 ppm, which is the top part, to the center, which is around 5 ppm. If you recollect, in the previous slide we saw the center is water. So, typically below water means downfield of water comes aromatics and mides, but upfield above water comes the aliphatic, and that is what is shown here, and you can see each amino acid has a very distinct pattern of peaks expected in this region. So, for example, glycine, of course, there will be 2 peaks, it can be 2 peaks or it can be 1 depending on the chemical shift, but it has only 2 alpha protons. There are no hydrogens in the beta, gamma, delta, it is only alpha, and that is what comes around 3.5 to 4 ppm. Then comes alanine, which is what we have been seeing. There is an alpha proton which is around 4 to 4.5, or sometimes it can come at 3.5, but it comes in this region, and there is a beta proton which is very much up and comes around 1.2, 1.5. Then comes valine. Valine, as we just now saw, has alpha, beta 1, gamma 2, and that is what has a pattern like this. So, like this, for every amino acid there is a kind of a distinct pattern, which very easy based on those patterns to recognize what are those amino acids. Now, if you see here, these have only beta protons. You can see all these amino acids, cysteine, tryptophan, phenylalanine, tyrosine, histidine, similarly aspartic acid and asparagine. So, these are called AMX spin system because they have only alpha and beta protons, and the spin system terminology we have not seen in this course, we can see that later in the part of the course. And then these amino acids like glutamic acid, lysine, arginine, isoleucine and so on, they have beta, gamma and delta protons as well, and therefore, we use the word long side chains. But as far as our interpretation of NMR data is concerned, we will not worry about these nomenclatures, we will simply use directly the word. So, one has to know what does each amino acid stand for, and this is a single letter code, that is histidine, this is tyrosine, phenylalanine. So, I would recommend that you go through any standard textbook in biochemistry to know what letters we use for which amino acids. So, now in the next class we will focus on, given this nice peak pattern of each amino acid, how can that be used to assign the first step, which is spin system assignment, and then subsequently the second step of sequence specific resonance assignments.